one thing I find really fascinating is how cells know what to become. So when you replace the cells in your tissue, somehow they have to know what they're going to become. It's a very interesting study of cytology and histology. We're going to talk a bit about atrophy and about tissue um, regeneration. And we did see some intercellular junctions in the videos that you watched the other day. So um, I'd like to just cover those again. So this is a nice slide. It's in the lecture, so you'll find that on the website, also in the textbook. And it shows the three different kinds of intercellular junctions. Inter means between one thing and another, so two different cells, inter. If you're saying intra, that is within the cell. So the word intra means within, inter means between. So intercellular junctions. So there's the tight junction here, and it holds the cells very closely together. There's the desmosome. Here, looks a little bit like a zipper. And you can see that there is a gap in between the cells. It does hold the cells together, but it does not keep ions and water out. And here is a gap junction, which is more like a pore, really, whereby fluids can flow freely from one cell to another. And those are the kind you would find in muscles and in the heart, so that the muscles can beat synchronously and the heart muscles can beat synchronously. All cells really except blood are anchored to each other or to the matrix that is surrounding them by intercellular junctions. So tight junctions completely encircle the cell. They encircle it, joining it to surrounding cells. They're a zipper-like pattern. They're complementary grooves and ridges, and they prevent bacteria from passing between cells. It's particularly important in the brain, for example. They create what's known as the blood-brain barrier. And they're also very important for the blood-testis barrier. So in the blood-brain barrier, of course, you don't want pathogens and toxins from entering the brain. So the epithelial cells in the brain are very tightly packed. And the blood-testis barrier, it's the sustentacular cells. So the sperm must be kept separate from the blood because the immune system doesn't recognize sperm as belonging to the person, so it will attack the sperm. So it's separated by what is known as the blood-testis barrier. And that is the sustentacular cells forming these tight junctions, keeping the sperm separate from blood. Found in the gastrointestinal tracts and the urinary tracts as well. Desmosomes are like patches between two cells, holding them together against mechanical stress. So there's a gap between the cells, and that has a mesh of filaments. It doesn't encircle the cell. You'll find it in places like the uterus, the heart, and the epidermis, places that tend to stretch mechanically. So this keeps the cells from splitting apart. Gap junctions are also known as communicating junctions. So this is where cells need to communicate with adjacent cells. They're a ring of 6 transmembrane proteins. They form a water-filled channel, the gap junctions. They're found in embryos, they're found in cardiac muscles, and in smooth muscle. I'd like to distinguish between 2 different kinds of glands, endocrine and exocrine glands. Endocrine glands have no ducts. So endocrine glands secrete product into bloodstream. such things as hormones. Exocrine glands, however, have ducts. And the ducts empty their contents onto the surface of either the body or possibly into the gut. So I have a question histology file on the website. So if you're looking for the histology slides, they are on the Biology 1120 page. So that's just the very top page. If you click on that, you'll see the calendar, you'll see the outline of the course, and below that, you'll see all of the lectures and lecture summaries. So thank you for that question. If you haven't been able to find them, that's where they are. Okay, so it could be that these particular slides at the very end aren't in there, but they should be. But if they're not, I will post them. I will post them. And of course, you'll be able to see this online because I'm recording this lecture as we go along. And glands in general are mostly epithelial tissue. Those are the tissue that line cavities and that line ducts and that line glands. There are some mixed organs. That may have both. They may have both. The liver, for example, secretes bile into a duct. Into the bile duct. But it also secretes albumin into the blood. That's a protein. The gonads, that's the ovaries and the testes, they release gametes through ducts, but they secrete hormones into the blood. The pancreas secretes digestive enzymes into a duct, but the pancreas secretes hormones like insulin and glucagon into the blood. So those are mixed organs. Here's an exocrine gland. This is the structure, often formed into lobules. The stroma divides the gland. It's a combination of the capsules themselves and the septum. The septum are the tissue that separates the lobules. The other part of the cell is, or the other part of the tissue is known as the parenchyma. And those are the cells that synthesize secretions. Of the gland. The acinus or the acinus, that is a cluster of cuboidal cells that surround the duct. And as you can see, they have secretory vessels that contain the product that will be released into the duct. There are different kinds of exocrine glands that I think Dr. Campbell did talk about. There's the simple coiled tubular exocrine gland. This is a sweat gland. This is the secretory portion. And this is the duct. That's a sweat gland, and it releases its contents to the surface of the skin. This is a compound gland, compound SNR, like a mammary gland. So it has several lobules. This is a compound tubulo-SNR gland. For example, the pancreas contains those kinds of glands. So there's different kinds of glands. So how do glands secrete their product? Well, we can divide glands into various categories depending on their secretions. They secrete different things. A serous gland, for example, will produce a very thin, a very watery secretion. So a serous gland includes the sweat glands, milk glands, tear glands, and digestive glands. A mucous gland produces glycoprotein, that's a protein with a carbohydrate attachment, and mucin, it's called mucin, sorry. And the mucin absorbs water and it becomes sticky. And that sticky secretion is called mucus. So mucous glands secrete mucus. Mixed glands can contain both serous and mucous cells. The cytogenic type of gland releases whole cells. So it will release sperm and egg cells. The cells of the testes release sperm, and the cells of the ovaries release eggs. And there's variations in methods of cellular secretion. For example, a holocrine gland, which is very interesting, the secretory cells, they disintegrate. And that's how they deliver their accumulated product in some cell fragments. So the oil-producing glands of the scalp are an example of that. Holocrine. Merocrine glands release product by exocytosis. So that's just another way of classifying glands. Exocytosis. So in that case, the product is in a vesicle inside the cell and it gets released into the external surface. Apocrine glands are like merocrine glands, but they have a little bit of a strange appearance. The cytoplasm is not lost. That it used to be thought that cytoplasm is lost, but that is not the case. Mammary glands and the sweat glands in the armpits, those are considered apocrine glands. Mucous membranes are found throughout passageways open to the exterior. So your digestive passageways, these are open to the external environment. Respiratory, the urinary, and reproductive. All of those passageways are open to the environment. And mucous membranes consist of a mucous coat. So those are the glycoproteins, the mucin, a cilia. So here are the cilia that keep beating even after you die. The mucin is excreted from a goblet cell. That's the goblet cell. These are pseudostratified epithelial cells, and we'll be looking at those in the histology lab today. The basement membrane holds the epithelial cells, attaches them onto the underlying tissue, which is connective tissue. In this case, it's loose connective tissue. And that layer is known as the lamina propria. And that's underneath the epithelial layer. Underneath the lamina propria is the muscularis mucosae. So that is the smooth muscle that, if it were surrounding the intestines and the digestive system, would be the ones performing peristalsis and moving food through the system. Mucous membranes. There are other kind of membranes in the body. Cutaneous membrane is skin. It's your external body surface. It is stratified squamous epithelial tissue resting on a layer of connective tissue. It's relatively dry. It serves a protective function against water and pathogens. Synovial membranes line joints. The only connective tissue comprises that layer, and they secrete what is known as a synovial fluid. It's rich in hyaluronic acid into the joint, and that maintains the fluidity of the joint and lessens the friction between the bones. A serous membrane, the serosa, is the internal membrane. It's simple squamous epithelial tissue. And areolar tissue, it covers organs. It covers organs, it covers the walls of the body cavities, and produces a serid fluid, a thin, watery fluid. The serous membrane, called the endothelium, lines blood vessels in the heart, and the mesothelium lines the pleural, pericardial, and peritoneal cavities. That's the lungs, the heart, and the abdomen. Can tissues change? They can change from one type to another. That's called differentiation. So unspecialized tissue in the embryo, they become specialized. So there's some tissue in the embryo called mesenchyme. It's not functional in the embryo, but it differentiates into muscle tissue during development, which of course is functional in contraction. That's differentiation. Metaplasia is a changing of a mature tissue to another. So in adolescence, before puberty, a simple cuboidal tissue exists, but it changes to stratified squamous tissue after puberty. And tissue can also grow, of course. Hyperplasia is tissue growth through cell multiplication. So just through mitosis, and cells divide and they make more cells and more tissue. Hypertrophy is enlargement of pre-existing cells. So when you're, for example, increasing your muscle mass with exercise, with the weights or whatever, you're not getting more cells, more muscle cells. Rather, your muscle cells themselves are growing in size. Neoplasia, that's new growth, is the growth of a tumor. It could be benign, so non-cancerous, or malignant, and it's through the growth of abnormal tissue. Often these are in the form of cysts that can be easily removed. But any abnormal growth or any lump or cyst or anything unusual should definitely be checked out. What about tissue shrinkage and death? Well, atrophy is the name given to the shrinkage of tissue from the loss of cell size or number. And that does happen with age, senile atrophy due to aging, but it can also happen if you don't use your tissue. So if you're not using your muscle tissue, your muscles will get smaller, your muscle cells will get smaller. Necrosis is the pathological death of a tissue. So due to some kind of disease or some kind of disorder. Gangrene is due to an insufficient blood supply. So sometimes alcoholics will get gangrene because the blood vessels of alcoholics tend to get thinner and more difficult to supply with blood in their extremities. Gas gangrene is due to anaerobic bacterial infection. Infarction is the sudden death of tissue from lack of blood. That's the myocardial infarction is the coronary arteries that are lacking blood. Decubitus ulcer is a bed sore or a pressure sore. So that's where blood does not reach the area due to pressure on the blood vessels. Apoptosis is programmed cell death. So that is genetically programmed so that the cell is disintegrated and macrophaged and essentially self-digested and then phagocytized by white blood cells. It's not due to inflammation. When you're an embryo, you have webbing between your fingers, but those cells die through apoptosis. And the uterus after delivery experiences apoptosis, as do earlobes. Tissue repair. What about when your tissue gets damaged in some way? Well, it occurs in two different ways. Regeneration is the replacement of dead or damaged cells with original cells. It restores normal function. So when you have injuries to the skin can regenerate. Even the liver can regenerate. I have a friend who he was the husband of somebody I went to grad school with, and he was working at Science World at the time. Now he's a policeman. But anyway, he's always telling funny stories. But he thought that he could harvest his liver and sell it because it would just regrow again. So he was going to sell like a third of his liver every 6 months or something like that. Of course, he was just kidding. He wouldn't actually do that. So regeneration is normal replacement, but fibrosis is the replacement of damaged cells with scar tissue, which is collagen. It holds the organ together, but function is not restored. So muscle injury, sometimes scarring of lung tissue and tuberculosis, or the healing of very severe cuts or burns of the skin. Keloid is healing with excessive fibrosis, so that raises shiny scars. So if there is a laceration of the skin, the damaged blood vessels will leak blood. We saw some of this in our Khan Academy video. Mast cells leak histamine, which dilates blood vessels. That increases blood flow to the area. You might think, oh, you don't want blood flow to the area because you don't want to lose blood. But with that blood flow comes your white blood cells, which you need, of course, for the pathogens, the bacteria or viruses that are coming into, excuse me, the injury. Yeah, so it increases blood flow and permeability. And plasma from the blood seeps into the wound, but it carries antibodies, which we need, clotting factors, which we need, and white blood cells. All of those things are instrumental in repair and wound healing. A clot forms. So the blood clots, it coagulates. That will form a scab on the surface. And inside, macrophages will start to clean up the debris from the injury. Fibroblasts will grow or will secrete rather collagen, and leukocytes will clean up also debris. Then new capillaries grow into the wound. Fibroblasts will deposit collagen, and this phase begins about 3 or 4 days, but it can last up to about 2 weeks. The surface epithelial cells will multiply and spread beneath the scab, and then the scab will eventually fall off. So the scab was there to keep other pathogens from entering the body, and of course from your body fluids from leaking out, but eventually your epithelial cells will multiply and spread beneath that scab. The epithelium grows thicker as it regenerates. Connective tissue forms only scar tissue, fibrosis. A complete remodeling, depending on the severity of the injury, may last up to 2 years. Excellent. Thank you for your attention.